We're going to shift gears now and talk about what some of these drugs are. Okay, so that soapbox talk was about telling you what you can do so you will be the recipient of an investment by third parties who've never had a family member affected by TM, NMO, or ADEM, who are not here for emotional reasons. They are making business decisions, and we are trying to make us the right business decision. And there are things out there that we could bring into this community today as we organize and get ourselves put together. We're going to talk about two this afternoon. I'm going to give the first, and the second is actually going to be presented by one of our industry partners. But this talk is on an agent called Anti-Lingo. Just by way of disclosure, this is a product that is patented and owned by Biogen IDEC. And we've had collaborations with Biogen IDEC for a long period of time. But I'm going to give you a little bit of the story of this drug, how it came to be, and then what the opportunities are.
Before we transition, I've got one question related to the previous presentation. We are a small target, and I understand that, but what about joining our efforts with the general focus on getting drugs for remyelination in general that are outside of our target that expand the base of potential utilization?
So great question. So what Don was asking is, we're a small group, but how can we leverage us as a beta tester guinea pig, I'm paraphrasing, for other demyelinating diseases? And the big gorilla in the room is multiple sclerosis. That's a huge market. It's become in the top 2 most expensive diseases in the United States in terms of drug costs. There are massive investments that occur because it's a larger market. And one of the things we want to do is showcase the fact that testing, and I'm going to talk about it in this talk, showcasing TM, NMO, and ADEM is a great way to deleverage risk for going to the bigger market. That we could be used as a stepping stone, but then we get all the benefits to our community. And so that's one of the strategies we talk about, and it's actually specific to the agents we're going to discuss. And I'll use that as an example. It's a very good question. So the lingo of no-go. There are parts of this talk that's going to be a little painful for everybody. So this is like getting Hannah to eat her vegetables. You're going to have to sit. You're going to have to think. Rebecca's sitting there. That's my eldest from many years ago, contemplating. She's actually pooping in the picture, but which is sometimes what happens when we think too hard. And we're going to put this in the context of translational medicine. Has everyone heard the term translational medicine? It's kind of a catchphrase, gets used by NIH. Let me tell you what it is. So at its core, translational medicine is about understanding disease. In order to understand disease, you need interplay between at least 3 different groups. So you need understanding of clinical medicine. I need to know what happens to you, what your symptoms are, what you're dealing with, all of these different issues. You need interplay from human biomarkers. I need, frankly, samples and specimens or imaging, which is a biomarker, from you, about you, to interplay what are you experiencing and what's happening in your body, the structure and the function. And then we need people in labs from basic science to either on their own generate hypotheses or take things from you to drive hypotheses. So sometimes in research there's a scientist in a lab who's never heard of neuromyelitis optica, who discovers a mouse who looks kind of weird, and then we realize the mouse kind of looks like neuromyelitis optica, and it drives the engine. Sometimes it's a clinician who says, I'm seeing this in a certain patient population, you really need to study this. Sometimes it's a blood sample taken from humans that discovers an anti-aquaporin 4 antibody, and then it drives the basic science. You can start understanding disease at any point, but at some point, all of those things need to talk to each other in order to better understand the disease. And that's translational medicine, bringing things out of the lab and into the clinic and vice versa. The other way to do this is to basically say, you are at the center of everything. Understanding you is the goal, and you need a lot of people on the team to understand you. You need the clinician, you need testing, and you need PhDs or MD PhDs or MDs in a lab doing research at a cellular or molecular level. And all of those things need to come together in order to better understand you. Now, for your concerns, this is what we're trying to get a grasp of. You've seen this picture a few times over the last 2 days. Everybody can now, I'm going to turn it off, you draw it, you have to label the dendrites, cell bodies, axon, myelin sheath, synaptic terminal. So the dendrites at the top are the interconnection between all the cells. The axon projects to the target. That insulation around the wire is there to make sure the signal gets from point A to point B, and the synapse is the connection. So if this is a motor neuron, and you have a motor neuron that's 1 meter long, going from your brain all the way down your spinal cord, and then another one, single cell that's a meter long, that goes from your spinal cord to your big toe. And that neuron has to go from the spinal cord and then synapse on the muscle. Lots of things can go wrong with your nerves. And you heard from Dr. Cheng and saw those great videos and pictures yesterday. I was watching all of you. For those of you who weren't here, we got to see some videos from surgery on nerve transplants. And I was watching around trying to see who put their head down and who got up to walk out. Nobody walked out. Everyone did well. But he talked about the fact that nerves can regrow and can regenerate, but this is something that happens in the peripheral nervous system. So you have a nerve, and the nerve is myelinated by cells called Schwann cells in the periphery. So the nerves that go from your spinal cord to the muscles have Schwann cells that make the myelin. And if you damage that myelin, these macrophages, these cells come in and clean it all up, and then the nerve can start to regrow. And if it doesn't make it, Dr. Cheng can come in and try and help out in different ways, but in general, there's actually regrowth. Every month, a little bit at a time, the nerve regrows. That's not what happens in the central nervous system. So in the central nervous system, when you get damage, the cells that make the myelin are now oligodendrocytes. So two different cells making myelin, one in the periphery, one in the central nervous system, but they're there to wrap up that axon. And when you get demyelination of the axon in the central nervous system, the debris gets cleared, but the nerve doesn't regrow. It just sits there or dies back. So what is it about the central nervous system that inhibits this, and why would you even want, biologically, why would your body want to inhibit growth of neurons in the central nervous system? And this probably comes from development. So we're all in utero, we get born, we make myelin over a couple years, and our brains are making connections. Synapses are forming. So if you start to develop visual systems, first off, babies just see black and white. They don't see colors. So if you go into a baby store, how many parents here recognize that for the little things you put above a crib, all those toys for the first month, they're all black and white because we figured out that's what kids see. If you do colors, they don't see it. And then it's over months that they get those synapses forming and firing and myelinated, and you start to have color vision. So from a development point of view, once you start to make connections, the brain wants it to stop. They want a certain amount of ingrained connectivity, not constantly changing. In order for you to form long-term memories, how many people here can remember the name of their kindergarten teacher? So mine was Mrs. Tenney. She hated me. When I graduated medical school, I'm sitting at dinner with my family, and my father says, well, if Miss Tenney could see you now. I said, well, what do you mean? And my mother says, Steve. And she goes like this. I said, what? She's like, well, we never really told you. And I said, told me what? And if you ask my brothers the story, the way they tell it is Miss Tenney thought I should be in special ed. The real story is Miss Tenney felt like I shouldn't be in an advanced gifted and talented program, or I needed to go into the next grade over. And so I wanted a copy of my med school degree sent to her. They wouldn't do it. But I remember her vividly, and the reason I remember her vividly is the synapses formed, they got hardwired, and that is important for our development. So there is a reason to have a stop signal in the central nervous system. This is not a bad thing biologically. It's only a bad thing when things go wrong. So what do patients teach us about nerve injuries? So the first is that patients with peripheral nerve injuries can show signs of repair and growth over time, but patients with central nervous system injuries have little evidence. And so the question is, what stops the repair, and can I inhibit that inhibition? Can I get rid of the stop signal? So by a guess here, we did the cost of drugs. For how long has this been recognized or studied? So it's 2013. We're finally figuring out inhibition. How long have we known about this process? 10, 20. So it's over 100 years. So Santiago Ramon Cajal, is Carlos in the room? Carlos, this is for you. This is for you. He doesn't, I would argue that Cajal is one of the most under-discussed, absolutely pioneering neuroscientists of the last 200 years. We talk about all sorts of individuals. He won the Nobel Prize in 1906 for his work in hand under very primitive microscopy showing synaptic trees and connections between neurons and recognizing the fact that a damaged central nervous system neuron does not regrow in 1900. Late 1800s. And the pictures are gorgeous. You go back to his books, and what's really creepy is they're incredibly accurate. We can take out microscopes today, and I can see, or Carlos can see, exactly what he was seeing. things have gotten a little better since his time. And it's really over the last 20 years, now 30 years, that we really started to understand the mechanism that inhibited growth. So he recognized the fact that there was inhibition, but now we're starting to tease apart. And there are two major inhibitions in the central nervous system to regrowth. The first is the glial scar, the astrocytic glial scar. And I forget, somebody asked me this question during one of the breaks. If we can repair things, what are we going to do about the scar tissue? Scar tissue is a problem. Okay? So those astrocytes, as they embed in a spinal cord or in a brain, they can inhibit the growth of neurons. And we're going to talk about what the possibilities are. The second, and here's a funny one, myelin inhibits neuronal growth in the central nervous system. So if you can get neurons to grow in the spinal cord, often what they'll do is grow up and down the spinal cord, but they won't leave. The myelin on the outer part of the spinal cord prevents them from branching out and trying to reconnect. Very unusual. There were a series of antibodies that were generated against different proteins within myelin that when you put them in, they turn off the myelin stop signal. So there's something in the myelin that's a big red stop sign, but if you can hide it, then all of a sudden the neurons will grow outside the spinal cord. And this is the work that started occurring in the 1980s and 1990s. If that wasn't enough, it gets a little more complicated. So in biology, for every signal, whether it's a stop or go signal, there's a receiver. Okay? So if I tell a neuron to stop growing, there is something on the neuron that catches that signal, that hears it. It's like the receiver on a two-way radio. And often in biology, there's more than one signal for each receiver and more than one receiver for each signal. So when we start to tease apart at a molecular level, why do some things connect and not others, we have to take into account all of these. And that's where the really fun words come in. No-go, lingo, mag, OMGP. It's like a foreign language as you go through these. But I listed for you some of what we know. So the stop signals within myelin are a group of proteins. One of them is called no-go. Great name, no-go. That's literally what it is. Don't go here. MAG, which is a myelin-associated glycoprotein. The oligodendrocyte myelin glycoprotein is OMGP. And then there are receivers for those. So on the neuronal side, there's something called the no-go receptor. And then P75, NTR, really fancy names. The interaction between the two, the summation of it, is what allows the cells to talk to each other, to either allow for growth or not allow for growth. But it turns out, so there was a lot of work that went into this, but it turns out these weren't the only proteins. So with the Human Genome Project, we found out that there's about 30,000 genes at least for humanity, and the overwhelming majority of them, we don't know what they do. Okay? So we have the code, we can predict a protein, we look at the protein and we're like, well, maybe it does blank. But until you actually test it, you don't really know. And there was a protein in there called Lingo-1. There's Lingo-2, 3, and 4. But Lingo-1, which was a protein that was predicted to make a sequence that was predicted to make a protein in the central nervous system. And when they did studies, sure enough, what you're seeing here is if we take pieces of brain, colon, heart, kidney, your whole body, and we take little pieces out, this Lingo-1 was just in the brain in this study, and we didn't see it anywhere else. So it's a protein that's specific to the brain. And when you looked at the structure of it, people way smarter than me said, well, geez, maybe based on its configuration, maybe it has something to do with this no-go pathway. And sure enough, when you put it into the mix, what you find is that this lingo attaches to these other proteins on the neuronal side, and it impacts the stop signal. So this was the first piece of data coming out, and this paper was published in 2004, looking at the fact that this pathway, the lingo pathway and the lingo receptor pathway, was part of the grow, no-grow signal. So what happens if you block it? All of a sudden, you actually start to see growth occurring of the neurons. So you can turn off the inhibition signal on the neurons, and you can see the neurites in the central nervous system start to grow out. So it becomes very attractive. And in fact, if you do this in animals who've had a spinal cord injury model, and you measure how well they can walk, their hind paws, and you track them all over the cage, if you inhibit lingo after a spinal cord injury, these rats repair much better. They regrow and they start walking far better than the natural state of repair. So there is some that happens, but it hits a stop point. If you inhibit this over a month, they do far better than the control. And there's a reason they recover. When you stain their spinal cords, what you're supposed to see are growing axons, which are little blue dots here. And if you injure the rat up here and you don't give them anything except water, you don't see the axons. If you don't injure them, they have nice axons there. If you inhibit lingo after the injury, they look pretty close. Not exactly, but pretty close to normal. So the axons are able to grow if you can turn off the biologic stop signal. Their capability is there. They're not dead. They haven't gone away. They just need to get the right coaxing in order to grow. But that's on the neuronal growth side. What about the remyelination side? So it turns out that Lingo-1 has an effect on the oligodendrocytes. Who here remembers what an oligodendrocyte does? Makes myelin in the central nervous system, which is different than the Schwann cell, which makes it in the peripheral nervous system. And right now you feel like Hannah eating the vegetables. If you block Lingo-1 at the oligo level, it makes more myelin. So the red is myelin, and if you have oligodendrocytes in a dish and you don't do anything to them, they have a little bit of myelin being grown. But if you turn off Lingo in the oligodendrocytes, all of a sudden they make myelin like gangbusters. Well, that's great. Making myelin's good, but what we really want the myelin to do is wrap around axons. Just to have a big glob of myelin in anyone's spinal cord isn't really going to do much good. So if you mix these together with cells, with neurons, with axons, you get these nice red myelinated axons, these lines. So it doesn't just grow myelin willy-nilly. It will implant and wrap the axons of the neurons by turning off this signal within the oligodendrocytes. And so when studies were done, and these were published over the last 5 to 6 years, in a model, EAE is the mouse version of multiple sclerosis. Okay? So you give a mouse an insult to their immune system, you coax it along a little bit, and all of a sudden their immune system will start attacking their spinal cord and brain and causing demyelination. If in these mice you go in and you give them anti-lingo, their oligodendrocytes will go in and remyelinate. What you're supposed to see here are shades of gray, not black. Black means no myelin, so this was a control animal, and this was one that was treated with anti-lingo. So their injuries were exactly the same, but if you get the oligodendrocytes making more myelin, they will go in and they will repair. So that's taking things from a dish, from the cell, taking it to an animal. Now, what's next in those phases of trials? Remember, there was the preclinical, then phase 1, 2, and 3. So now it's time for humans. So Anti-Lingo-1 is currently in clinical trials. Here's the update. So the first trial looked at both healthy volunteers and individuals with relapsing remitting MS and progressive MS. And this was purely a safety study. So the phase 1, it's good to know that there were no serious adverse events. They didn't get any signal that said you need to stop the trial. And they have moved on to phase 2. Now the phase 2 study is in multiple sclerosis, and it's currently going on in Europe. I'm sure they'll be in the US as well. But this is progressing along. Now when it gets time for companies to start talking about phase 3, it'd be really nice, and these discussions have been going on for several years now, to consider TM, NMO, ADEM, and we'll talk about why in a moment. But it'd be something that we're gunning for because there's a lot of opportunity here. But now we got to go. So the trials are happening, but now as our community, we have to go back to the basic science. So we got to go full circle again. So remember this slide? All the different things that can inhibit, and we talked about the lack of myelin. There's one other thing that can inhibit, the glial scar. To date, I don't know of any data, Mike or Carlos may know of some, that says the Lingo pathway would do something for a glial scar. I'm not aware of any data to suggest that it would. It's more on the neurite growth and the remyelination side. So how would basic science knowledge about a glial scar and how important it is impact trial design? And this is where our community comes in. So in TM, NMO, and ADEM, the honest answer is timing of therapy for these types of interventions is going to be important for the remyelination side. I think what you're going to see are designs that push for people to be in trials within the first year or two after an injury, maybe even the first several months after an injury, so that the confounding factor of a glial scar is less of an issue, just on the scientific side. Duration of therapy is going to be a consideration. How long should we give this for? We don't want these cells turned on forever. We need the outcomes to be measured. We have to separate patients based on how much axonal pathology or demyelination. And this is where a study like what Lindsay Horton presented earlier becomes incredibly important. Because if we have a capability in our community to tell the greater world, I can separate out demyelination from axonal pathology, and I can find patients whose functional deficits are from demyelination, naked wires, they become the best candidate for a remyelination strategy. And companies appropriately and scientists love to enrich a population. So that study that Lindsay did has huge implications for our ability to design a trial and recruit the right patients into it to give the most bang for our buck. But we have to sort through a lot of these things within our community to make us the attractive recipient for these types of phase 3 trials. And it brings it back where we started on the translational medicine side. You have to understand the disease. You get there from the clinical side, you get there from the biomarker side, you get there from the basic science side. If we do this as a community, we are setting ourselves up quite nicely, whether it's anti-lingo or something else. Why would I advocate, to Don's question, for TM, ADEM, NMO to be the prototype diseases to test remyelination strategies? Well, the first is, for TM and ADEM, it's a single event. It's the hit and run. And so if we want to quantify repair, I don't have to worry that there's another event coming later that would damage what we just repaired. And that's a very attractive quality about patients with TM and ADEM. NMO is equally attractive for a different reason. NMO patients can have relapses, but they don't have a progressive side to their disease for the most part. 99+% of NMO patients. In MS, they're secondary progressive disease. And so now, if you're trying to study remyelination, you have to account for the fact that even though you remyelinate, patients may still look worse year after year after year. In NMO, it's discrete events. So at the time of enrollment, if they don't have relapses, you can quantify the remyelination effect. These are very attractive qualities about you scientifically and biologically that we need to harness to put the spotlight on you for remyelination strategies. What we need to do, however, to make this even better is the last two. Defining the outcomes in natural history studies, we need to know what happens to you. We just need to be able to sit at a table with the FDA and stop talking about a third, a third, a third. Because that's what they're telling me, and I have to kind of hang my head and say, well, that's not really true, but we can use it for now. And then ideally biomarkers, things from your blood, from your MRIs, from your OCT, from your spinal fluid that tell us, are you going to be a responder, non-responder? Is there something about you that would make you a good candidate? These are ways that we can invest in the grassroots level and get us where we want to be. So where I'd leave this is the following. Remyelination therapy is absolutely coming. It is not science fiction. It's not a pipe dream. It's not 20 years from now. It's real time. It is going to be here. You need to be ready, and you need to be ready in two ways. You need to be ready as a community, organizing to make sure we make ourselves the attractive recipient for these resources. And you need to be ready as individuals. This goes back to Carlos, and he said it the best. What can you be doing now? Exercising, 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 PT, PT, PT. Because guess what? If you have a contracture, or if you have spasticity that's affected your muscles or your ankles, or if you have secondary osteoporosis, or if you've developed sequelae from disuse, you are not as attractive a candidate as an individual for remyelination strategies as somebody who has kept in tip-top shape as best as possible, has functional deficits, but when their spinal cord repairs, we're going to see the benefit because their muscles are loose, they've been doing their yoga, they've been exercising every day, they've avoided the complications. And so the investment you make as an individual is extremely important in making sure that you as an individual are a good recipient for these types of strategies. We're going to do our work trying to get them ready. You have to do your own work to be a good candidate. All right. Thank you for your attention.